quick reminder, please, if you have not contacted me yet to meet with me for a product, please do so as soon as possible. I realize the due date is on Monday. You can turn in your form, and I would like to meet with you as soon as possible after that. So a lot of you have already contacted me, and I have a lot of you on my calendar next week. So make sure those of you that have not sent me an email yet to do so with your availability. So availability in any given week based on your class schedule, every possible time. Do not ask me for one particular time. I do have a crazy schedule this semester. So give me all possible availability, and then I'll find what works. Okay? Good. So before we start a cool topic, which is chromatography, I just want to go back a little bit to previous lecture and have some recap. What is a sampling plan? Okay, John. What is everything? Yeah? So the first thing you need to put in there is your objective. What are you trying to accomplish with your sampling plan? So your objective or the purpose of the sampling. The second most important thing is to figure out how to withdraw a representative sample. Okay? And then after that, then you can, decide on, mention the methods, mention how you are going to prepare the sample, store the sample, etc. So it's like your working document. Sampling plan is your working document that starts with objective. Okay, why is a sampling plan important? Okay, Sonia, go ahead.
It's important because if you don't take samples well, then it can throw off all your further analyses.
Exactly. So no matter how excellent of an analytical person you are, no matter how your precision is great, your accuracy is going to be thrown off if your sampling plan is off. So basically you start there and then move on. If you screw this up, we're done. What is the difference between attribute and variable sampling? Okay, Ben, the end there. Okay, and there's one important difference between attribute and variable. Whatever you said is correct. But what is a very important difference of attribute and variable in terms of distribution? Yes, Sven? Yeah. Yes. So the attribute is like it could be in one corner of your bag, it could be on the surface, it could be on the bottom. So it's not really homogeneous, and that's why you require a lot of sampling. What is a composite sample? Farah? A one composite of multiple samples. Okay. According to FDA regulation for nutrition labeling, how many samples should be drawn to form composite samples, and how many subsamples? Jenny?
Isn't it you need 12 to form a composite sample and then 6 subsamples?
6 subsamples, okay. When is it hard to perform probability sampling? Can you think of a time where you cannot really do probability sampling? Ben. What's that? Okay, so experience, okay, but not sure about the wine aspect of it. So there are restrictions sometimes, Sarah. That's a restriction right there. Is like you cannot reach other places. So yeah, that's an example. When you cannot get sample from a big compartment. When is it, well, that's, I asked that. what are some sample preparation considerations? So when you're preparing the samples, what do we want to watch out for? Can you list some? Okay, Vaidi. A representative sample, okay, but there are other kind of, while you're preparing the samples, Aaron? Limit enzyme activity. Farah, do you have anything else? Lipid oxidation. Christina? Never mind. Okay. So any alteration to the sample, you want to make sure that you avoid. So you don't want microbial growth, you don't want enzyme activity, you don't want lipid oxidation. You want to make sure you label your samples correctly. You don't want to mislabel. Storage conditions under not high humidity or high temperature. When you grind your sample, you want to make sure you grind it following the best procedure so that you don't lose moisture, you don't overheat, etc. You get the right mesh numbers and size of your particles. Who didn't get this sheet that I was passing? Uh, Everybody got? Do we have one missing still? I did print 34. Anyway, this is a summary table for chromatography studying that is not in your slides, so I made a printout. If you didn't get one, there's one more here. Okay. Chromatography. It's the best topic ever. We're going to spend about 4 lectures on basic principles of chromatography. All right. Who can define chromatography? What is it? Yeah. So nobody can, didn't you learn about chromatography in any of the previous chemistry classes you've taken? Okay, so I see heads nodding. All right, so what is it? What do we use it for? Just simple. Okay, Bailey. Yeah. So you have 2 phases and you're separating things in your sample. So you have the sample and it has so many different components in there and you want to separate them so that you quantify a particular component or quantify all the different components. You have a mixture of antioxidants in your sample. Example for Amy here. She wants to analyze the content of all of her hydroxycinnamic acids that she extracts from intermediate wheatgrass. So she needs chromatography to separate them into individual components so that she can quantify them. So does anybody know why it's called chroma? Chroma? Yeah. Yeah. Yes, chroma is coming from color. So one of the 2 first scientists that discovered chromatography, and actually the one that got credit for it, is a Russian scientist. His name is Sven. Not you, Sven. And he was a botanist, and he separated color, the plant color, into separate colors using not earth, chalk. So, and then graphy, because you graph your results. So that's where chromatography comes from. So since we're talking about history a little bit, so actually the first person that ever used this concept was actually an American geologist. His name was Day, and like night and day, and he separated crude petroleum using fuller earth. So that was the first person that ever used chromatography, but then Sven described the principle and gave it the name chromatography because he separated colors from plant, and he got the credit for it. So, and then after that, it got forgotten for a bit, and then other scientists came along, discovered partition chromatography, which we're going to talk about today or next week. And then later on, gas chromatography in the 50s evolved. And few years later, high-performance liquid chromatography. A few years later, in the 60s, supercritical fluid chromatography evolved. So we're going to talk about all the different types of chromatography, the different modes of separation. So when I tell you what type of chromatography, I would be asking really, is it a gas chromatography, or is it a liquid chromatography, or is it a planar chromatography, paper chromatography? But when I ask you what's the mode of separation, how are they separated with this type of chromatography, then I'm going to want you to tell me it's adsorptive chromatography, it's ion exchange chromatography, it's reversed phase chromatography. Okay, so I'm throwing so many names at you right now, but all of these we're going to talk about in so much detail. After 5 lectures, you're going to be experts in chromatography. So I always tell a story about this and about chromatography just to simplify it in a practical manner. When my daughter was 10, 11, or 12, now she hates it when I tell her this. She's embarrassed by it. When she was that little, she used to love to go to a store in the mall. And it's a very girly store, lots of pink and suffocating purple. And it's called, no, not Claire's, Justice. So let's say myself and my daughter, we want to separate. The best way to separate us is take us to Justice. Because I go in and I want to come out right away. I don't want to interact with any stationary phases inside that shop. However, she goes in and goes around and takes her time. She gets delayed. So I elute before she does. So I leave, I go with the void volume, and you learn that the void volume is the volume that goes through and nothing interacts with the column. So I elute with the void volume. However, she takes her time, interacts with stuff. The only way I can get her out is buy her something. So that's the mobile phase, where in order for something to leave the column, you have to give something in the mobile phase to have that solute come out. So basically, this is how it goes. So that's chromatography. You have the stationary phase where your solute or whatever is in your sample is interacting with the stationary phase. And then the mobile phase is carrying you through, but in order to come out, you have to give something in the mobile phase to help come out. Okay? So whatever the mode of separation is, there are going to be different stationary phases and there are going to be different mobile phases to help separate whatever you are separating. So this is just an overview, but we're going to go about and talk about everything in detail. Okay. Knowledge check, and I don't expect you to know this, but let's see. So like I said, there's going to be an interaction between a stationary phase and the solutes, whatever is in your sample. This kind of interaction is intermolecular interactions because mostly you're separating molecules. So if you have adsorption chromatography, which we'll talk about and describe, what type of intermolecular interactions do you think is going to happen between your solute and the stationary phase? Adsorption chromatography. So you have these listed. Any idea? Any wild guess? Bill. B and C. Anybody else? Okay, let's see. Agnes, yeah? Go ahead. Okay, Agnes, you are correct, all of the above. So in absorption chromatography, any of these or all of these may occur. And today we're going to learn, or not today, within the next few lectures, we're going to learn which mode of separation is going to use electrostatic interaction most, hydrophobic interaction, hydrogen bonding, and in one type of chromatography, actually you use all of them. There will be all of them occurring. So And there's one other one, which is van der Waals interaction. So reverse phase chromatography. Have you heard of this term before? Reverse phase chromatography. You might have, and if you have, you might have forgotten. And if you didn't hear about it, we'll hear about it in class. But any wild guesses here? In reverse phase chromatography, which compound will elute or leave the column first? Michelle W. A and C. Yes, you are correct. Woohoo! Yes. In reverse phase chromatography, your stationary phase is generally nonpolar, and your mobile phase is a more polar. And with that, because the mobile phase is polar, what's going to happen, the polar compounds will interact more with the mobile phase, less with the column, and leave first. Okay? Again, these concepts, we're going to repeat them as we go along. Okay, chromatography is an extension of a concept known as extraction. So in extraction, you are transferring a solute from one liquid phase to another, or even sometimes it's solid. It doesn't have to be liquid. But if we want to think about extension of chromatography, let's just think we have two liquids. Let's say we have an aqueous phase and we have an organic phase. Let's say something is dissolved in water, and then you have hexane is an organic solvent. So the solute that you're after might like water better or might like hexane better. So if you take your sample and you shake it in a compartment that has the two phases and you wait until they settle, you will have a phase separation because hexane is immiscible with water. Then your solute will tend to be in the phase that it likes better. It's more soluble in better. So most of the concentration of your solute will be in that phase. So that's extraction. You're actually isolating something out of a complex matrix. So extraction can be an actual mean of analysis. Can you give an example? You'll learn about that during the entire semester, but can anybody think where sometimes you do extraction and you get a quantity of something? Fat extraction. We're going to do Soxhlet analysis in lab for fat determination. So we use extraction to determine fat content. Okay, preliminary sample cleanup. So an example of that Sonia is very familiar with.
Protein extraction.
So what do we do before we extract protein? If we have a product that is high in something? We defat. Yes. So if you want to extract protein for characterizing of that protein, and if your matrix is high in fat, the fat is going to interfere with the extraction of protein. You have to remove the fat first. So a preliminary sample cleanup is that example. And then concentration of the component of interest. This could be like in flavor chemistry, and if you want to quantify a certain component that is present in really small concentrations. So you have to extract them, concentrate them, and then you can analyze them. So there are going to be a lot of examples the entire semester on extraction, use of extraction, and use of chromatography as well. So I'll point them out as we go along in the semester, but for now, these are just examples for you to get the concept. So there are different types of extractions. You have something called batch extraction, where like the example I gave you, this is a separatory funnel. And here you have, let's say, an organic solvent, and then you have an aqueous phase, so you can see clear separation. So if you're extracting something that is in your aqueous phase and you want it to go to your organic phase, you're going to do something like that when we do the modiner, for example. There are going to be two immiscible solvents in there. We're going to use different type of funnel or flask. But anyway, in this case, you shake and you let it settle and leave it as such. That's batch extraction. So it's not a continuous process of extraction. Success, I don't know what's happening today. Success extraction is a continuous. Actually, in fact, it's a semi-continuous. And you're going to learn about that when we cover the fat chapter. But basically, you have a solvent will evaporate. Your sample is in a second compartment. And you have a condenser. And then this is on a heating block. So your solvent will evaporate, go up, and then get cooled down by the water running, which is the condenser system, and then drops back into the sample. And if you're doing that with ethyl ether, for example, then you're extracting the fat. So then it goes in, solubilize the fat, and then after you reach a certain level, it siphons back into the solvent with the oil. So the solvent carries the oil and bring it back down, and the cycle continues. And we learn when we do success is that it can be as low as 4 hours, and sometimes, depending on the sample that you have, 8 or 12 hours that you leave it there to extract your fat. Now, countercurrent extraction is what the extraction that led into partition chromatography. So one thing I want to go through before going to countercurrent extraction is there is something called equilibrium. So when we shake and let it settle, the solute is going to equilibrate. Let's say 90% of the solute will be in the organic phase and 10% of it will be still in the liquid, in the aqueous phase. So with that, when you get that first equilibrium, you're going to get a concentration of the solute in one phase, let's say the organic phase, and then the concentration of the solute in the second phase. At equilibrium, this is a constant. The ratio of the concentration is a constant, and that constant is called partition coefficient. So this is very important because chromatography is all about equilibrations. So separation happens between the stationary phase and the mobile phase by a series of equilibration. And at every point, at every equilibrium, you have a specific partition coefficient. Okay, so now going into countercurrent extraction, I used to have this link that takes me to a demonstration applet. They call it applet. But Java no longer is supported by the Firefox or Explorer or whatever. So if I click on it, it doesn't show the video anymore, unfortunately. But it shows the guy, okay, see? It shows this guy who discovered countercurrent chromatography. Oh, you can't see it. Why can't you see it? Okay, how do I get out of presenting? I did. Doesn't want to escape. Oh. It's not escaping. Oh dear. Help? TA? Support? How do I get what? Okay, and he's giving suggestions here. Can I duplicate my screen?
Yeah.
Yeah, I don't know why it's not duplicated.
Display settings.
Oh, this is a new it's new program. It's so annoying. Do you know, Andy? Oh,
Here, this is why. Leave it there and
and
then
extend, right?
duplicate.
Duplicate,
Yep,
apply.
there. Your website.
Keep changes.
Yeah. Now,
If I escape, yes.
yeah.
Ha. So there used to be a video or an applet here, but it cannot be displayed anymore. But anyway, this guy discovered that if he puts tubes in a series, it's like this kind of tubes in a series, and in a lower compartment of the tube he put one liquid, let's say water, and then from one end he introduces hexane, let's say, okay, immiscible. So, and then he puts the sample in the first tube, and there's water, and then hexane, and then there's a shake, 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 and then the content of the first tube is is spilled into the second tube. But however, the lower part of the whatever was in the tube, the water, remains in the first tube. So only what gets spilled into the next tube is your organic phase. So basically what he's doing, so each tube is like that. So you have water in here, and then you add your second solvent, and then a shaking occurs, and then the solvent at the top, which is your mobile phase, goes to the second tube, basically. And this is repeated multiple times. The more tubes you have in a series, the better separation you're going to get. So basically, this became partition chromatography later on, became the principle of partition chromatography. So if I go back here, so here is the very primitive extraction tool. There is a hand cranking thing that you do the shaking and then the spilling. And then this was the applet that I used to use, but it doesn't work anymore. But basically it shows how your component, different colors, you have the green and the pink, how they move. One is more retarded than the other. So not retarded mentally, just retarded in terms of time and movement. So you look here at the coefficient of separation here. So A is 0.5 and B is 3.5. So let's say A is more soluble in the stationary phase and B is more soluble in the mobile phase. So which one will elute first? So you have a smaller coefficient here and a larger coefficient, and then the B is more soluble in the mobile phase. So which one will move faster? B. Okay. So here this means that A dislikes the mobile phase more than B does. So if I want to illustrate it in a peak format, this is what this is what you will be seeing, peaks coming out of your computer program when you run chromatography. So what happens is you have here, let's say we're measuring absorbance at 340 nanometer. And this is time. If I have colored pens, I would have drawn the green and the blue, but I'm going to say you start seeing this is your B moving faster, coming out of your column faster, reach a peak in concentration, and then your A will follow, basically. And this is a peak because you have a certain concentration and they don't all come out. Not everything on the column is going to come out at the same second. So they're going to be coming out until most of them are out, and then more and more is coming out until nothing is coming out anymore, and that would be your peak. Peak area, usually we correlate to concentration. And then the same for B. And when I talk about, when I go over column chromatography, you'll get that concept even better. So we'll come back to that later. So yeah, unfortunately it was a cool applet, but unfortunately it doesn't work anymore. Um, but anyway, the countercurrent extraction extended into partition chromatography. And partition chromatography is basically where you have a stationary phase, and let's say you have a column, you pack it with a certain material, and then you have a layer of a liquid bound to that column or to that support material, and then you have a liquid, a mobile phase that goes through, and then there will be equilibrium between the liquid that is bound to your support material and then the liquid that is going through the column. So that was the basis for it. So you're going to hear a lot of terms. Oh, by the way, like I told you last, I don't know when I told you this, I do update my slides, and this in particular, this chapter slide has been updated a little bit more than others because I base it on the revised chapter that will be published next year. So, so I posted on Moodle my updated slides. So you can get them there, and the table that I passed out is going to be in the chapter next year, but you don't have it now, and it's small font on the slides. That's why you got a handout for it. Okay, so you're going to hear a lot about these. You're going to hear these terms come up, and I want to make sure that you know what they are. Solute would be anything that is soluble in your solution, that is in your solution, and you're after separating or collecting it. Stationary phase, by definition, stationary, so it doesn't move. It just sits there, accepts your solutes, interact with the solute, and then your mobile phase is the moving phase that carries your solute with it. And then your mobile phase, you're going to hear eluting solvent. So the mobile phase can also be called eluting solvent. Eluting because whatever comes out of the column is your eluent, comes out of the column. And what's eluting it is the mobile phase. So, and also eluting carrier. Carrier gas, when it's a gas, it becomes the carrier gas. And then you have a supercritical fluid. So you have a solvent, supercritical fluid, and a carrier gas. So you're going to hear that depending on what type of chromatography you're doing. And then series of equilibration, that's what I said. At a certain equilibrium, you have a one concentration in the stationary phase and another concentration in the mobile phase at equilibrium, and there will be a series of that within a column. And chromatographic peak is this. It's a chromatogram of a certain separation that you've carried. Okay. So chromatography is used a lot in food analysis, a lot. And we're going to have designated chapters where there are going to be applications on the chromatography that we're going to learn about in these few lectures. So application is going to come later on in the semester. So let's say carbohydrate analysis when we do sugars, mono, di, and oligosaccharides. We're going to learn about specific types of chromatography that will be used for that application. So it's basically a separation technique that we use so many different places: mycotoxins, pesticides, amino acids, enzymes, all sorts of different components that you can think of. It's a highly robust tool for separation and quantification. Okay. And I did say that earlier. It's based on the partition or distribution of your solute between a stationary phase and a mobile phase. And there is your partition coefficient or distribution coefficient constant at each equilibrium. Okay. So these are the different types of of chromatography or methods. Okay? You have gas chromatography, you have supercritical fluid chromatography, and you have liquid chromatography. So the lectures that I'm going to cover is mostly on liquid chromatography. I'll introduce these two, and there are going to be 2 lectures on gas chromatography separately with Gary Renexis. Okay? So in liquid chromatography, there is something called planar chromatography. Planar, because it is a flat plane. So it can be vertical or horizontal. So planar chromatography is paper chromatography and thin layer chromatography. Paper chromatography is ancient. And how many of you have done paper chromatography in chemistry? You should have all have done that at some point. Thin layer chromatography, some of you might have done that as well. I will talk about each one. They're very similar, but thin layer chromatography has better resolution. And then there's column chromatography, which is the most common chromatography used for separation and quantification. Gas chromatography is also a column chromatography because there's always a column involved. And then in gas chromatography, you have gas as your mobile phase and liquid as your stationary phase. Your slide shows gas solid. Cross that out. No longer. So nowadays, gas chromatography, your stationary phase is basically a capillary tube. There is a capillary tube. Your column is a capillary tube column. That means hollow in the middle. And then you have a support lining within that column. And then you have a bonded liquid phase. The bonded liquid phase is your stationary phase. And then the gas, you have a carrier gas, is your mobile phase. Oh, I missed something here. Oh, in paper chromatography, it is mostly partition chromatography, liquid-liquid. When you have a liquid mobile phase and a liquid stationary phase, this is called partition chromatography. And that's the chromatography that stemmed from counter-current extraction chromatography. Counter-current extraction. All right, and then column chromatography, where you have a column involved with a stationary phase packed within the column. You can have, again, your stationary phase is a bonded liquid phase to a support material, and the mobile phase is always liquid. Hence the name liquid chromatography. And then you can have a solid stationary phase with your liquid mobile phase. So these now, we're talking about different modes of separation. So we talked about types, but now these are modes of separation. On what basis your solutes are separating, or what concept? So you have size exclusion, which is based on size or molecular weight or molecular structure. Weight and structure can play a role in separation. And then you have the partition chromatography, where you have a solute that is soluble in one liquid more than the other. And then you have adsorption chromatography, where your solute interact with the solid via intermolecular interactions. They adsorb on the surface of your stationary phase. Within, whether it's partition or adsorption chromatography, whether you have a liquid stationary phase or a solid stationary phase, you can have 2 separate modes of separation: reversed phase and normal phase. In normal phase chromatography, I think I have it on a separate slide. In normal phase chromatography, your stationary phase is polar and your mobile phase is nonpolar. And it is called normal phase for one simple reason. It was discovered and used before the reversed phase. So first they started doing polar stationary phase, nonpolar, and then they swapped. They discovered material that they can have it nonpolar as stationary phase, and then your mobile phase became polar, so they just said reversed phase. They reversed it. Within adsorption chromatography, there are specialized adsorption chromatography, very unique and specialized. Hydrophobic interaction chromatography, you don't have it in your slide, I added that. So hydrophobic interaction chromatography, we'll talk about it. And then you have affinity chromatography, we'll talk about that, it's very specialized. And ion exchange is very specialized. You have to have charged molecules that you're trying to separate. Okay. Yes. Yeah, partition chromatography is basically you have two immiscible liquids. They don't interact with each other. Your stationary phase is a liquid phase, and your mobile phase is a liquid phase. But then you have solutes in your sample. They would partition. I mean, they will part, either more in the first phase or the second phase. That's why it's called partition, because some of the concentration will be mostly in one phase than another phase. Okay. This is a summary table you'll find in your current textbook. It's very good as a studying table, so that it kind of gives you the method, the mobile phase and the stationary phases, and then how does the retention, again, this is in, I should have added to the terminology. Retention means how long it stays on your column or get retained on the column or on the stationary phase. So, and then you'll hear terms like retention time, the time it stayed on your stationary phase, or it was retained on your stationary phase. So staying on the stationary phase is affected by different properties, and for each method you have different retention differs by the different properties. So we'll go over all of these in more detail, but this is a summary table that you want to come back to and make sure that you know. So very brief introduction to gas chromatography. Gary is going to spend 2 lectures on that, but I just want to introduce it really quickly. In gas chromatography, as the name implies, your carrier phase or your mobile phase is a gas. So here's the system. So you have a big black box, and in this box there is a column. This tube right here is a capillary column. Capillary columns are very, very common in gas chromatography, and they're very, very long, so for better resolution, that is. So the longer the column, the better resolution. We'll talk about that later. But it's coils because it is very long, so, and it's flexible, so it coils. So when you open the black box, you're going to see a column attached that is coiled. So the black box is actually an oven, okay? So it heats up your column. And here you have several tubes, gas tubes. You need to make sure that they're hooked to the system if you want it to work. So you have a carrier gas, which is your inert gas, is the helium. Does not interact with anything, but it is your actual mobile phase. And then you have a source of oxygen, can be oxygen gas or can be air. And then you have the hydrogen, and both of them needs to be connected to your system. Especially if you have a flame ionization detector, okay, which you will learn with Gary, the FID. Anyway, and then you have a place which is called the injector where you inject your sample. So this is very brief. Gary's going to go over each compartment separately, but what happens is you are separating thermally stable volatiles. So you're looking at components that get volatilized when you heat. So then they can move with your gas. If they're not volatile, they're not going to move with your gas. And if they are not stable to heat, they're going to degrade and you won't be able to detect them. So basically what happens is the gas is going to carry your sample that is volatile, and that sample is going to start interacting with the components of your stationary phase. And then the separation occurs. And you can build a gradient based on temperature. So you can raise the temperature over time, and then they will evaporate based on their boiling point. So the separation is basically based on their molecular size, their polarity. Usually the columns, you have non-hydrophobic columns, and then their boiling point, at which temperature they will evaporate and be carried with the gas. So after they elute or leave the column, there's a detector, and then from there you get you integrate using a computer program and you get a chromatogram. So this is just an introduction. Like I said, you get 2 lectures on that. Any questions? I'm wondering if I should stop here because I won't finish in 1 minute. Do you have any questions? No questions? I'll stop here for now. I'll continue on Monday. I'll see you in lab, Monday group. Have a great weekend.